You wouldn't want them to another video conference instead of Good. Are you ready for some rain? Some more. Yumalundi, everyone, and welcome to the IAE seminar for this week. For those of you who don't know me, I'm Professor Ross Thompson. I am Director of the Centre for Applied Water Science and Co-Director of the Institute for Applied Ecology. And I'd like to begin today's seminar by recognising that we're on the lands of the Ngunnawal people, never sold and never ceded. I'd like to express my respect for their elders, past, present and emerging, and recognise that over on University Hill there is evidence that for the last 30,000 years people have been meeting to share knowledge on this site. And so we are on a site which has a great responsibility and also a great deal of value as a place of exchanging knowledge. And so the seminar today will continue that long tradition. Today's speaker is Peter Unmack. I really wanted to construct this incredibly ornate and completely fake introduction to Peter, where he was like born into abject poverty in the slums of Rio de Janeiro or something. But his actual CV is still interesting, but not as cool as that. So Peter was born into the slums of Melbourne and was brought up in Victoria, and from a very early age, from his teenage years, was motivated to go out and work on Australia's probably understudied freshwater fish fauna. That led him on to a Bachelor of Science at La Trobe University before doing his Master's at Arizona State, which is where he picked up the slightly odd accent, which makes everyone think he's American. He's not. It's a fake. Then he went on to a PhD at Arizona State University. He's worked extensively on native fish on every aspect, from describing species through to field mapping, biogeography, natural history work, conservation ecology work. He's published over 100 papers in that area. Most recently, he's been really heavily engaged in using genomic and genetic technologies as tools for freshwater fish conservation, and he's a genuine leader in this space. He has until recently been a senior research fellow within the Centre for Applied Water Science, but now he has reached out and formed his own company, which I've currently forgotten the name of.
Unmack Aqua Evolutionary.
Unmack Aqua Evolutionary. And so he's clearly your go-to guy for all your fish conservation needs. I'll pass over to Pete.
Thanks, Ross.
And I've bucked the trend where in the last few years, probably more 5-6 years ago, everyone from Monash moved to University of Canberra. I've actually just started a one-year part-time postdoc there at Monash Uni just to buck that trend. You can slightly blame Ben Kefford for this talk today. So ever since Ben got here, every time semester series comes around, he's always, hey, do a talk on your carp gudgeons. I'm like, piss off. I don't want to talk about carp gudgeons. Too much work, So I finally have gotten a talk together. And so hopefully it lives up to his expectations. Thanks, Ben. So today we're talking about hemiclonal carp gudgeons. Oh, we're not moving forward here. There we go. Now, there's really two key people in this project, right? So nothing ever gets done by one person, right? So this is a much broader collaboration with tons and tons of people. So Mark Adams has kind of, I guess, been our godfather to Michael Hammer and I through all of our fishy research. He's really the brains behind all this stuff. I just go out, catch fish, genotype them all, and he actually figures out what it all, the most important bits of it. Mark's been a fantastic collaborator. I've known him since about 1989. He works on everything under the sun, but we've really done a huge amount of fish stuff together. So he's an Alzheimer jockey, which now his lab's folded up and he's retired the Alzheimer stuff, but it's always been a fantastic complement to whatever we were doing. That key thing of getting the nuclear data combined with the mitochondrial data. That was just having mitochondrial data is a big weakness. So he really strengthened massively everything we did. Michael Hammer, I've known Michael since about 2001. He's a mad keen fish collector, and he's currently based at the Northern Territory Museum. He started out in Adelaide, and he's had a really long-term interest in carp gudgeons as well. And we pretty much, the three of us collaborate on pretty much everything on Australian fishes together. There's always lots of other things to acknowledge. We've had multiple funding sources from ARC through the IAE, the Czech Academy of Sciences, some stuff through the Natural History Museum of Geneva. Dozens of people have helped me with field work over the years. I'm always trying to rope anybody I can to go out and hold the other end of the seine and abuse for a few days. So key ones are Mike Boltzley, a mate from Arizona, Carl Moy, who's here, Tim Page from Griffith Uni, and my wife, Rochelle, who's here today. And of course, there's been lots of other academic collaborators and all kinds of other projects, Jonas from the eDNA lab, Tariq, Chris Hardy from CSIRO down the road, Matt McGee at Monash, Susanna, Peter Rubb from the Czech Republic, Eloise from France, Sweden, Dan Smith, who did some of the early work at Griffith University, and Chris Thacker from LA County Museum. And of course, I suck at taking photos. All the photos are stolen from somebody else. So for some reason, when I was about 8 or 9 years old, I bugged my mum in Kmart to buy me a fish tank. And I don't know how many times I'd bugged her, but one day she finally bought me a fish tank, and I've been keeping fish pretty much ever since. The best place to put fish tanks is by your dinner table, so that when your wife talks too much, you can sit there, watch the fish, and actually pay no attention to anything she's saying, but she still thinks you're actually listening. Or maybe she doesn't, but she talks anyway. But you get to watch the fish, and it's really wonderful and peaceful. I've been very heavily involved in the hobbyist side of the aquarium trade through the Australian New Guinea Fishers Association. I'm actually currently president of that group, and I've been heavily involved in the production of their journal, Fishers of Sahul, the last few years as well. I got into angling when I was probably about 11 or so years old as well. That's the first redfin I ever caught. And just by random chance, that's my father and grandfather behind me, which was just purely photobombed accidental. I was really, really lucky that my parents always have traveled a lot within Australia primarily, usually in a car and stuff. Dad bought a four-wheel drive in 1985, wanted to go to Flinders Ranges. And I found places that were interesting to go collecting fish, and they were happy to come along and camp for the weekend or school holidays or whatever. So we went all over Western Queensland and Northern Victoria and all over the place. Back when I was, 15, 16 years old was when that kind of started. And of course, my current family, Rochelle, she's held the other end of the seine quite a few times and copped her fair share of abuse from me when we can't catch the fish. And of course, my two little monsters who aren't smaller than me anymore, Trevor in the middle and Andy on the right. Andy's now taller than me, and they're both going fairly feral as teenagers do, but they do their own little bit of fish collecting as well and pull the seine occasionally and manage to do okay. The beautiful thing with this sort of career, you get to travel a lot, right? So, I've caught fish all over the place, and this is just a small snapshot of the places I've been, but I've killed a lot of fish in my time, and I'm very proud of it. But the beauty of that is we have massive genetic and morphological collections that we've built up over this time, and that's been really critical for all kinds of things. and will be critical, going forward 50, 60, 100 years from now. These collections will still be highly valuable and useful. So in terms of the sort of things that I'm interested in, it's basically anything to do with fish. But essentially kind of my academic career got a bit more focused on biogeography, phylogeography of freshwater organisms. How do they move between river basins? Being based in the US, it's a bit hard to do any field-based, ecology-based stuff. So the biogeography, genetic stuff was a really good way of bridging that gap. So I'm interested in all aspects, the biology, conservation, systematics of Aussie freshwater fishes. There's a lot of interesting questions around, people go dump fish in a river for conservation or fish stocking. No one's really actually investigated how we make that sort of stuff more effective. There's a lot of work to be done around that sort of stuff. I've always had a massive interest in desert springs, especially edgebaston and the redfin blueye, which was a species I was lucky enough to discover back in 1990, and also interested in figuring out ways we can kill gambusia. Now, I first started keeping carp gudgeons in 1985. Basically, we're just up in the Murray River. I had a little aquarium net. You go dipping around the billabong, you catch these little fish, you bring them home. That's kind of what I did. The best book of the time said there was 3 different types in the Murray-Darling Basin, but it was pretty quickly obvious there were 4 present. So that kind of started off my early interest when you're just a teenager. Hey, there's a fish in here that's not in the book, and it's clearly different. So eventually I kind of published all my natural history observations in 2000 in Fishes of Sahul. Just a bit of a summary of what I'd bred, my observations on distribution, all that stuff. Now, Mark Adams had been mumbling about carp gudgeon hybrids and some weird funky stuff for about a decade to me. And I was always just like, he doesn't know what he's talking about. It's like, I've never seen any of this hybrid crap, whatever. And of course, being Mark, he was right. So this was their paper that came out in 2000, which was the first one to really document this system. Now, the key part with this system is, right, so the key part here is so that for all of these things, we use codes, HA, HB, HX, and so forth. So basically, you've got these things that are HA and HB, which are nice sexual species. There's a little cluster of hybrids between them. But then you've got these HX, X just meaning, X was unknown, right? And so that's a classic signal of a hemiclonal system where one of the sexual parents is absent, right? They've gone extinct, but the hemiclones persist, right? So they didn't have evidence proving the system was hemiclonal, but it had all the classic signatures of it, which was beautiful. And at that point, we're still kind of like, okay, there's all these hybrid things out there, but it was still a bit kind of mystifying and stuff. And of course, this wasn't Mark's first rodeo, right? So he'd done some earlier work on heteronodia with Craig Moritz and Dunellen and Baverstock. This is only one of only 2 times you'll ever see me show a lizard in a presentation. That's the beast that they were working on. And then later he did a whole bunch of stuff on Manettias as well. And that's the last lizard you'll ever see me show in a presentation. Now, the origins of stuff. So we always kind of separate unisexuality in vertebrates versus invertebrates, probably just because it's simpler that way, and who the hell knows what goes on in invertebrates because they're even crazier. But the first fish record was Hubs and Hubs in 1932, the famous Amazon molly. That was the first record of this in a vertebrate. It had been recorded in invertebrates prior to that. So yeah, we're only talking 90 years of knowledge around this sort of stuff. And so they've always fascinated biologists, though, in a big way. This is a really excellent book by John Avis, who only writes excellent books. He's a fantastic author and really good at summarizing stuff in a very intelligent and understandable way. But this book kind of goes through everything you would ever need to know in a very understandable manner about unisexuality in vertebrates and and it's got a few other things as well, but it's a really good summary. So why do we care about unisexual vertebrates, right? So sex is almost universal, but it has a massive cost, right? The problem is you produce the perfect genotype, say me, but as soon as you mate with somebody else and make kids, you've diluted that perfect genotype by 50%, right? And that doesn't work very well. But, we all do this, and that's just the limits on how it all works, right? Whereas if you're parthenogenic, you can just clone yourself and you've got perfection for, until you die because you don't have any genetic diversity. So unisexuals are very uncommon. There's, you hear a a bit of talk about them, but that's completely disproportionate to how common they are. But they provide really excellent opportunities for studies, especially ones that can be brought into the lab and manipulated in that manner. They're generally seen as evolutionary dead ends, and there's not many examples of old taxa that exist. There's a couple of invertebrate ones that actually go back something like 100 million years, possibly, but most of the vertebrate ones are maybe in the order of hundreds of thousands to maybe a couple millions of years kind of time frame. It's hard to date these things. Nearly, I say most examples arise through hybridization. I think it's probably virtually all of them do. Sometimes it's a little bit unclear. Males in these unisexual vertebrates are incredibly uncommon, right? There's only 3 or 4 groups that have males, and most of the time males are very, very rare. And the carp gudgeons are the opposite. There's just as many males as females. So there's 2 major types of unisexuality: asexual and sexual. So the parthenogenesis is the asexual version. Females produce young with no contributions from male. You don't need anybody else. You just need a female. She can produce eggs and make more females. And that's predominantly found within a dozen genera within squamates, but it's quite common in a lot of invertebrate groups. There's 3 modes of sexual reproduction. The first is gynogenesis. This is what the Amazon mollies do. Basically, they need a male to fertilize them, but the sperm is not used. The sperm triggers the development of the embryos, but there's minimal, if any, contribution from the male genetically. There's a tiny bit of slippage sometimes, but essentially they don't contribute. So basically you need a male, but the genetic contribution is 0. So they're essentially, they're not parthenogenic, they're essentially clonal as well. Hybridogenesis, which is what the carp gudgeons do, is hemiclonal, right? So they basically borrow a genome from a species each generation. So essentially, if you've got a regular sexual one on this side, these are essentially F1 hemiclones. They always look like an F1. So you've got the red genome, which is a separate species' genome. And essentially, each generation they'll mate with the sexual guy, grab its genome, but then there's no recombination. So when these things make gametes, all of their gametes are the red genome. The blue genome gets excluded, right? And there's a few fishes that do that, and there's a big group of water frogs in Europe that have been pretty extensively studied that do this as well. But it's not particularly common. And then the third one's a weird one called kleptogenesis. So basically, in this group, what happens is you do get incorporation of new genetic material into those lineages. Usually comes about through adding entire genomes, changing the ploidy. And that's known in some salamanders, and they're currently considered to be the oldest vertebrate unisexual system. The hypothesis being that they get new genetic material from time to time, and that allows them to adapt and keep track of things. All right, so on to carp gudgeons. So basically, I'm just going to go through a few of the different aspects of the research that we've done over time, starting with carp gudgeon phylogeny and distribution. So the genus, like quite a few in Australia, likely have an Indo-Pacific origin. Within Australia, you've got one very widespread species, goes all the way around the coast. That's the empire gudgeon, Hypseleotris compressor. Then you've got this radiation in the northwest with one in the Pilbara, and then this big radiation here in the southeast, and really not much in the areas in between. And they go as far as from Japan to Madagascar, South Africa, so all through the Indo-Pacific. But there's probably only 1 or 2 species in that marine section, and then a few in the Pacific where there's a little bit more diversity. We did a UCE phylogeny. We've done old phylogenies with mitochondrial data and similar, but this time we did it properly, probably 500 loci or something or other. This was largely driven by Chris Thacker, and largely shows you've kind of got a monophyletic lineage here in the southeast, a monophyletic lineage in the northwest, with the empire gudgeon kind of stuck in the middle. And then the first branching lineage just represents the stuff across the Indo-Pacific. Now, the hemiclonal complex consists of 5 sexual forms. These are all in southeastern Australia. And depending on where you are, depends on who you can mate with and what sort of hemiclones are present. Until recently, most of these were undescribed. There's 3 what we call Gallii types historical, which we call HA. So that consists of the firetail gudgeon, the cryptic carp gudgeon, the Mary carp gudgeon. So that's represented by the guy on the top here. A lot more red in the fins, more elongate fins, slightly slenderish body. Then we've got what used to be called Midgley's carp gudgeon, which we've renamed the buffhead carp gudgeon. That's this one here, and that's for its big buffy head. That's what we always call HB. And then the bald carp gudgeon. So these guys are basically scaleless across the entire upper portion of their body and head. And that's HX, the missing sexual species. So these were described earlier this year with Chris Thacker, her husband Dan, and myself. So we described 4 species and redescribed 1. So the buffhead's now called Bucocephala, meaning big head. Cephala is head. The bald carp gudgeon is Gymnocephala for bald head. The cryptic carp gudgeon is Acropina for pointy fins. And the merry carp gudgeon is Mulubuluensis, which is a nightmare to say and spell. And then H. gallii was the existing species that had been described back in about 1910 or so. So the buffhead is the most widespread one. It basically goes from the southern end of the wet tropics throughout the deserty stuff, all the way down through the Murray-Darling Basin. The bald carp gudgeon is only known from 2 little tiny creeks in the upper Lachlan River, where it was fairly recently discovered. And then within the gallii group, you've got Acropina throughout the Murray-Darling and a little bit on the coast, gallii on the coast, and then this only occurs in the Mary River. It's not uncommon for the Mary to have different taxa in it to those surrounding it. It's a super important group in southeastern Australia, more so today, given most other fishes have declined and disappeared. They're super widespread, they're super abundant, they're undoubtedly an important food resource for fishes and other birds, and they're probably a really important invertebrate predator. And, you know, they can be ridiculously abundant. You know, this is one seine all, and we didn't count them, but there's probably, 2,000-3,000 carp gudgeons in this thing, in the dam after it refilled. So they can rebounce very rapidly from drought and other problems. Nearly anywhere you go in the Murray-Darling Basin, you can find gambusia and you can find carp gudgeons. They're pretty much ubiquitous. Now, historically in research, everybody's always just lumped carp gudgeons together as Hypseleotris because no one can tell these things apart. And they are challenging to tell apart, but that severely limits our understanding of their biology, right? Especially when it's one of the few groups left in the Murray-Darling Basin. If you want to, look at environmental health and that sort of thing, and you've just put three or four sexual species all into one bucket, plus all their hemiclones, you've lost 7 things that you're calling one in a system that's nowhere near as diverse as it used to be. Some of these are moderately easy to tell apart once you get your eye in. Others are incredibly difficult. And there's some I can't tell apart without the genetics yet. So basically, you can go out, pull the seine net in many places in the Murray-Darling Basin and catch 3 sexual species along with 3 hemiclones, all in the same seine net, all living in the same place, all apparently doing similar-ish things, right? Which is ridiculous. That's a ridiculous number of things to all have living together, persisting all together. There was an earlier paper that was done that modeled some of this stuff and it basically said these should all go extinct in a few generations. So there's clearly some really interesting aspects of their ecology that are enabling these things to persist and all live together. And there's about 4 people that can tell these things apart. I managed to train Carl, he got pretty good at it, but it's not easy to do. And it's really helpful to be familiar with how these things look in aquariums, which was how I first figured it out. When all you've got is dead fish, it's a bit challenging. And small ones are really difficult. But essentially, just to revise here, so we've got HA, which is part of the gallii group, which is 3 species in that, HB, the buffhead, and then the bald carp gudgeon, and then this is western carp gudgeon, Hypseleotris klunzingeri. So this one usually co-occurs with these guys, but they don't hybridize with them. But that's the sister group to this radiation here. And they've got very distinctive fin colorations and scale patterns and stuff, and they're fairly easy to distinguish from the others, even at tiny sizes. But it gets really hard when you get to the hemiclones of these things, right? So the top one's a gallii, this one's a hemiclone with buffhead carp gudgeon. So one little point is anytime you see the asterisk, that indicates the hemiclonal portion of the genome, right? So HB is the one that gets passed on clonally each generation. So HB is a little bit deeper bodied fish and a little bit shorter, stubbier fish. So if you look at caudal peduncle depth, so this guy's just a fraction smaller than that guy. Deeper caudal peduncle, narrower up here. Fins extend to the caudal peduncle, or to the caudal fin. These guys are not as long and a bit deeper bodied fish, a little bit less deeper bodied fish. But these are big adult males, which are, so again, I throw them all in a thing and knock them all out and euthanize them, then sort them. I can pick them into two piles, but it's sometimes challenging to know, okay, I know there's two things in here, but it's hard to necessarily work out which one is which. But pairing that with the genetics really helps. And then you get to females, and these are two different types, but to be honest, I'm not sure which one's which in here. But essentially, one type gets this kind of greenish tinge here, which the other one doesn't. And there's some other subtle differences as well. So there's still a lot more work to be done to try and distinguish these things. Gallii is quite common in the aquarium trade. They're often used for mosquito control and stocking in dams and things. And they get beautiful, nice, long, elongated fins. And the females are unusual. They have a black spot on their bum. And that's the diagnostic feature for telling them apart. So they're actually easy to tell apart. And most of their range, they live alone. So that makes it a little bit easier too. And again, females around spawning time get quite a diversity of color and are quite attractive. They'll get pinky, purpley, orangey bellies. And some of these markings here are quite important as to telling these guys apart. But there's a lot more work to be done to better quantify that stuff. So the first paper that really provided more solid understanding of this system was done by Dan Smith through Jane Hughes's lab, where they're working in Northern Victoria. A really neat study system. So they dived into this and found all kinds of crazy stuff that made no sense. Well, they did get to make sense of it, but really, this was kind of the foundation of what was to follow for the work that we did. All right, so finding a ghost lineage. So this started off with Chris Hardy. So essentially, when I first moved to Canberra in 2013, he's like, I know a population where there's pure HX. And of course, I was a bit skeptical of this at the time because we've genotyped lots of these things and we've never found one. So it's like, okay, so we popped up. So it's actually in Gunning in Meadows Creek. Sample there, it's like, holy crap, this is the only carp gudgeon here. And it turned out that he'd just by random chance, he just said, I was in there in the middle of the drought. There was just a muddy puddle in there. I just thought I'd catch a couple fish. And that's why we know about this. All right. So it's these bizarre little random things. And of course, he actually had been genotyping carp gudgeons and had some interest. So he'd followed through. So he was really the discoverer of this story. But essentially, you've seen this beast a couple of times. But essentially, from the allozymes and other stuff, we knew if we found this ghost, that's about where it would come out. And that's where it came out once we actually found it. So again, kind of normally these hemiclones are about midway between the sexual species involved in this complex. Now, HX doesn't live with any of these other things today. It only occurs by itself or with Western carp gudgeon. Again, it's lovely when folks walk in your office with some data. So this is Jonas's eDNA data from Blakeney Creek system. He was doing stuff looking at redfin invasion with pygmy perch. And he comes in and he shows me this figure and I'm like, this is not possible, right? And the reason is, so this is from downstream to upstream. And essentially his upstream sites have pure HX. And it's like, that's impossible. And of course we go up there, we collect the fish and it turns out, holy crap, these are pure HX. So we had a second population that was living up in this area. And as you can see, as you move downstream, they all get replaced by HB. There's some Western carp gudgeon in the system as well. But essentially there's a little gorgy section and they, for whatever reason, the HBs haven't made it further upstream. But that's the two populations we know of. And so when we throw all this into the phylogeny, we'd known the HX mitochondrial lineage was already present in hemiclones, so we kind of knew what was going on. But these are essentially the Lachlan fish here, which are multiple mutations away from all of the other hemiclonal mitochondrial genomes that are out there. So they've been isolated from the rest of its genome for quite a fair period of time based on those number of mutations. So they're not just, recently recreated fish from hemiclones, because none of the hemiclones have that same mitochondrial genome. So it only lives in two creeks in the upper Lachlan. It could well be in other places, but finding them is a bit of a challenge. And again, we've sampled pretty broadly all over the Murray-Darling Basin and have never found these things. As I said, the key thing is it doesn't co-occur with any of the other hemiclones, right? So the primary threat to this thing is other carp gudgeons getting introduced, which is a far more common thing than you might think, because every time somebody buys golden perch to stock their pond, the people that sell golden perch drag a net through the pond. There's usually carp gudgeons in there as well, and they just bag them all up and you get carp gudgeons and golden perch. So you're paying money for fish that you don't want, and then they get out. Droughts and more probably importantly, floods are probably the primary threat in these sort of habitats. They made it through the 2019 drought pretty good, but you can get huge floods that sometimes follow these from intense rains, and they're actually the key threat to these things. But they're pretty tough fish. They can bounce back pretty good from many of these things. And of course, there's lots of questions as to why it's no longer found with the other species, and what happens when it co-occurs with other members of the complex. So there's a whole huge pile of research questions in that little bit just there. And its conservation is a really key part of understanding this complex. We really don't want to lose these populations because it's going to be really important to understanding the evolution of the complex. All right, so the main genetic pattern. So we've kind of dived into the SNP stuff these days pretty heavily, but essentially just to refresh. So again, your hemiclonal genome gets passed on clonally, uses the genome from another species. So essentially we've got the 3 sexuals in each color, and then you've got hemiclones between the 2, with the HB genome being transmitted in this hemiclone and the HX genome being transmitted in these 2. Now, as to why it's the HX gets transmitted and not the other one, I've got no idea. We've always assumed hemiclones can only mate with sexuals, because that's generally how it works in other systems, but that's probably completely wrong. And we've always assumed that hemiclones can probably switch. So if you're in a population, this thing can probably mate with either a HB and make another one, or it can mate with a HA and make one of these. We assume, we don't know that for certain though, but there's probably a bit of swapping and stuff goes on. I always assume that everything we think we know about these things is probably wrong, because it probably is. So again, this is just the SNP stuff, about 8,000 SNPs, so HB, HA, the bald carp gudgeon down here. So when you do the mitochondrial stuff on these sexual things, they're about 7 to 10% divergence, which is probably 10-ish million years if you believe in molecular clocks. So they're fairly, the whole key trick to making this work is you've got to be different enough that when you hybridize, you don't just make F1 hybrids that back cross and intermix between the 2 species. So what has to happen is they mate, it somehow breaks the recombination machinery, but they're not so different that the hybrids can't survive and persist. And it may well be that many of these matings never ever make it. But 1 in 1,000 may have just the right combination, and then that allows it to go and then continue. We don't know. We haven't had much luck trying to recreate these things in the wild, in captivity, but we haven't tried terribly hard. Now, the key things in here is that these are very heavily male biased, and they all have the mitochondrial genome from this group here, because they're usually mating with a female from that group. These guys over here, the HA, HX, or HG, so from these guys mating with these, they're very, very heavily female biased. So in the earlier stuff, these were about 96%, these were about 98%. So these guys have both mitochondrial genomes present in them, meaning both that one and those ones. And then for these guys, they have both males and females. The ratio depends on what site you're at. Sometimes you get more females, sometimes you get more males, but it's often about equalish. You get both mitochondrial genomes in those guys as well. I think those are the key points from that. All right, so parasites. So there's always been a whole pile of questions around unisexuals and parasite loads and these things, and whether parasites eventually end up knocking them off, or diseases or those sort of things. I was really fortunate. I had this wonderful student, Eloise Rochette, who came to UC for a few months, and we went out and collected some fish and ground them up. So the real goal here was just to say, are there any differences amongst these things in their parasite loads where they're all coexisting together? If one species, are the hemiclones more infected or less infected than the sexuals? We really had no idea what to expect. So we went out to Walga, collected a few sites, because Walga is a really interesting place for carp gudgeons. There's Eloise on the Murray River. If you ever get the chance to work with parasite people, do so, because they're crazy people. They're great fun. But I mean, she sat there for a week in the lab in Walga, just staring at the microscope, grinding up fish after fish after fish, right? I mean, you pull out their organs, you grind it up, you pull out their eyes, brains, every little bit's got different parasites in it. And it's just amazing. It's just like, yeah, she was really awesome. So essentially, we got some, I thought they're a bit surprising results. Don't worry too much about everything in this table, but just to highlight the key bits. So our HBs, which are sexual, had about a 95% infection rate down here. So the PI is the proportion of individuals that were infected. Our, what we called Lake's carp gudgeon, so that's the hemiclone between HB and HX, they had a 93% infection rate, whereas our Western carp gudgeons over here, this is the sister to those groups, only had a 39%. And so in all these numbers, basically, Western carp gudgeons had bugger all parasites. Our HBs and our HB-HXs had loads and loads and loads of parasites. Huge numbers. Again, these are all living together in the same place, presumably with similar ecologies, and yet their parasite loads were insanely different. The other fun thing about working with parasites, well, we did a bunch of genetic stuff and made some trees. So just to highlight a few things here. So these, you know, this is a humongously long branch, right? So the way these parasite guys go, they sequence some really conservative gene. You pull in all the data from GenBank, from all the other related things people have done, and you find out every single one, your genotype is typically different because there's loads more species of these things than other things. So this is a really massively different thing where it's got kind of like a family name, but who the hell knows what it is? You know, here's another one that's different. This is a little bit different to the rest, a whole bunch of different ones up here. So you're not only, you know, discovering new things about parasite loads on the fish, you know, half the parasites you find are new lineages or families or genera, you know, it's just the amount of work done on these parasites is crazy. And so there's a huge amount of stuff, you know, that could be further done with this type of thing, looking at the parasitology of these guys to understand what's going on. And despite these guys having really high parasite loads, they still seem to perform fairly well in terms of, you know, they live there, they reproduce, there's loads and loads of them. But, you know, why these things got different parasite loads, right? There must be some differences going in ecology. Maybe they're feeding a bit different behaviors that the parasites aren't keying in on. Maybe they have different immune response or other factors. You know, that's a whole pot that we've opened that who knows where it goes. Chromosomes. So it's unusual at UC we have two chromosome people. Chromosome people are kind of a dying breed. But chromosomes are really actually fundamental to understanding a lot of the stuff we do today. You know, back in the 60s and 70s, chromosomes was often the only way we could dive into the genetics and genomes of these critters. So lots and lots of people did chromosome stuff. And then once mitochondrial DNA took over, the chromosome stuff kind of dropped away. And there's very few people that know how to do this. But it's really, really critical on a whole pile of fundamental levels now. But there's so few people doing this stuff. So we had this wonderful opportunity that Tariq arranged through his collaborator at the Czech Republic, Peter Raab, where Susanna, so Susanna's the one in the middle there with Carl, and this was a visiting student from Utah. So Susanna came a couple times and dedicated her life to staring down microscopes and squishing fish and things and looking for chromosomes. She was fantastic to work with. So the goals were to kind of look at, we knew nothing about the karyotypes of these things, right? So to look at karyotype variability, cytogenetic conformation of hybridogenic reproduction, and look at the mechanism of how the DNA gets eliminated when these things make their gametes. And oddly, this is the only vertebrate group of unisexuals that we're yet to find any evidence of polyploidy in, which is really quite bizarre. So maybe they're unusual in that respect. And some groups only have very limited evidence for polyploidy, but that's the one element that's actually missing in this group from all the others. Now, this crazy table here, chromosomes will drive you nuts because I barely understand them myself, but essentially, chromosomes are usually paired, and so you always get an even number, right? Well, not in these guys. So you get everything from 45, 46, 47, 48. I thought we had a 49 somewhere, but it doesn't seem to be. And you can't really have 47 chromosomes. It's a bit odd. Whereas so that's just in the somatic cells. Once you get in the germ cells, though, it's usually pretty good, and most of them even out to 48. I'm not sure why there's a 47 there. The chromosome people could explain that one. So basically, huge variation in chromosomes and some weird stuff going on. And some of the stuff with the odd number seems to be part of the chromosome fusing with another chromosome. So it's not being lost per se. It's just moving to a different chunk of the genome, which I don't understand how any of that works, and they still live and breathe. But they do. Now, this chromosome slides are always just to me look like somebody's thrown up on the slide and then they explain all this great stuff about it, right? So ignore most of the key thing to point out here is that you've got two colors, and the colors represent the different genomes. So basically, they work out markers that identify this genome, species genome versus that species genome. So in your somatic cells, you've got a nice F1 hybrid with two types, but when you get to the gametes, they're all one type. Same here, green and red, but all green. Red and green, all red, so forth and so forth. So confirming that when they're making their gametes, they're clonal. The other chunk gets removed. And then I don't fully know all of the great details in here, but essentially when the chromosomes do their stuff, the foreign genome gets excluded. Basically, they just get broken up. And so they end up unattached to any microtubules, which means they get excluded from the process. And then they get enclosed in micronuclei, and then they degrade due to autophagy. And so the arrows point at, I think these are just bits of the other genome kind of gradually getting pulled out and then chewed up and destroyed. These diagrams are always a bit of a mystery to me. All right, so the key thing out of all this stuff is we've basically come up with a bunch of enigmas, right? So just weird shit that these things do, basically. So I love catching fish. So the great thing of doing these things is you go all over southeastern Australia. It's actually, catching them's fun. Processing them in the field is not fun. I hate doing that. So basically we've sampled about 100 sites, 2,400 or so specimens. So basically you're seeing up some fish. Sometimes you get some interesting characters wondering what the hell you're doing. That's my hamburger there later. Now, I'm pretty blind, so I actually have to take off my glasses if I really want to see things. So basically, we catch all these carp gudgeons, we throw them all in clove oil, which nicely euthanizes them. So it's a wonderful compound. And then once they're all inactive, basically I just sit there, stare at them and separate them into piles. So you end up with this, right? So one of these will be Western carp gudgeons, one will be buffhead gudgeons, and one will be Lakes carp gudgeons. It's a very biased sample. We're not just randomly sampling the population. We're trying to make sure we get 10 fish of each type, equal numbers of males and females, so that we can use that to apply to morphology and stuff. So it's not just a random sample of, we want 40 fish from this population, whatever the 40 we pick out happens to be. So it's highly biased in that respect. So then we take off a little tissue sample, which goes into liquid nitrogen. And then the fish itself goes in an individually labeled tube that gets filled with formaldehyde. And then I make Carl, when I get home, dump all the formaldehyde out and transfer them in through water and into ethanol with about 6 changes. It's massively painful. Carl enjoys pain. That's why he likes hanging out with me. But basically, you need this to start to actually properly figure out what's going on and to make sure you've got specimens of known genotype to do the morphology. And that's what we based our descriptions on. So we're kind of at the point where we're starting to think we know something about these guys, but it really just highlights how little we know. So there's five enigmas currently identified in the group, right? So there's just bizarre stuff, right? So where's the sexual males, right? So one of the hard parts with carp gudgeons, most places you go, you catch lots of different types. So that obscures some of the stuff that's going on underneath. But occasionally you get to sites where there's only 1 or 2 types there, and that's when you start seeing weird stuff. But it's incredibly rare in the Murray-Darling Basin to find a place where you only get 2 types. Very unusual. So markers, allozymed, a huge pile of these. We've genotyped a huge amount. But basically, HA, there's lots of females, but then there's no males, no male sexual HA genotypes. And that's a lot of fish. And so again, heavily male biased. You might remember I said the HA-HB is very heavily male biased, but there's a few females there, about 10% of females are HA-HB. And then in Southeast Queensland, we've found the same thing where you get Gallii that co-occurs with HBs as well. And again, it's quite common in a number of these populations, no males. And a much higher ratio of females in those guys as well. So, you know, and there's no evidence of sex reversal in these things. You can normally in fish, if they sex reverse, you put a bunch of single sexes into an aquarium, and between a few weeks to a few months, they sex reverse if they're going to do it. I also had Catherine Hassel from Melbourne, did some of the histology on their gonads, and they don't have the bits you need to be able to sex reverse. So part of Dan Smith's work actually worked out that, you know, he had genotypes there where there was no sexual species present, right? So again, remember we said, you know, we often thought you needed a sexual there to mate with the hemiclones so they could persist. But here we had hemiclones with a genome that wasn't present in any of the sexual species. And that's, persisted long term. So it's clearly not just a fluky thing that he went there on the right day and there was some weird fish around. We've repeated this sort of stuff. So essentially he's got, the sexual species over here making more sexual individuals. And then he's got hemiclones with HX and HA, which keep mating back to these, we presume. And then we've got HA times HBs, presumably mating back to those, but there's no HBs in this system. So then what happens is we get those two hemiclones making these guys. So basically there's HB, HX fish in this system, but there's no HB sexuals, there's no HX sexuals. So they have to be being created from two hemiclones, different hemiclones mating together. Now we don't know, are they a dead end? Do they mate back to something else? No idea. And of course, normally in most places, HB is also present. So this could be going on in a lot of the populations, but we just can't see it because HB is usually ubiquitous. It's unusual to not have HB. The third enigma is around sex ratios, right? So there's some really funky stuff. And there could be a lot of different reasons why you get strange sex ratios. So in some times you get hybrids, for whatever reason, one sex is just incompatible. That's lethal. The two genomes just don't work together. So that way you can end up with a really biased sex ratio. And we don't know if that's the case in these guys or if there's something chromosomal or other stuff going on. But essentially, so these are Dan Smith's numbers here. So we got 98% HA, HB males and 96% females for HA, HX. Now this is from our 2,400 fish that we sampled, a very biased sample, right? We weren't just randomly sampling fish. But essentially, we get the same thing where oddly, there's a bit of a female bias in HA, and it's often common to find populations where there's very few male sexuals. We did a bit better than 98%, but again, we're highly biased in how we're sampling, but it was fairly close, slightly better than 96, but still males in this group are fairly rare. I call that fairly even on the grand scheme of things. And HBs, again, we're biasing this though, because we're trying to get equal representation of males and females. Tariq has a hypothesis that there could be an XYZW system working in conjunction in these guys, and that's a pretty bizarre idea, but that may possibly explain why you get these weird sex ratios going on. They're not really well known in vertebrates in terms of having two sex determining systems operating at the same time, but they have done it in lab-produced tilapias and other things and developed those systems. So whether that pans out or not, it's a little bit yet to be seen, but it's a really interesting thing as to why certain hybrid combinations only produce one sex and the other one produces both. Now, the fourth enigma is where you get into this kind of weird stuff where you get coexistence of multiple sexual and unisexual forms at the same sites, but the sexuals are quite rare, right? If these things are being driven by requiring the sexuals to be present to mate with the hemiclones, if the hemiclones are too successful, they essentially eliminate the sexuals through competition and other things. But clearly that's not happening. So here's Swanpool Creek, just a little tiny creek in the Broken River system near Benalla. We got 5 sexual individuals versus 21 hemiclonal individuals. This is the Severn River in Queensland and the Northern Murray-Darling Basin. Similar thing, you've got about 29 fish out of 100 that are sexual and all the rest are hemiclonal. And again, very, very few males in that system that have a sexual genotype. So pretty freaky. And Dan got the same pattern in his sites in Victoria where he looked at it. And then at 2 sites, by chance, we've found HX, but these are not the original sexual HXs that are in the upper Lachlan. These are ones that have been recreated essentially based on their mitochondrial genomes and a bit of other evidence. But the Wagga Wagga is a really fascinating place for carp gudgeons because there's just a totally bizarre and unusual pattern there that's not replicated anywhere else in the Murray-Darling Basin. So essentially we have huge numbers of HB, HX hemiclones, incredibly heavily male biased, so we caught about 100 of those guys. We only got 12 HBs of the sexuals, and then just out of the fish that we genotyped, we had 2 HXs, which have HB mitochondrial DNA. And then in Bite Creek, this is just a tiny little creek in the Kiowa system in northern Victoria. And here you've got no sexuals and 3 HXs, and they have HX mitochondrial DNA, but it's the normal HX you find in the hemiclones, not in the sexuals. And so they weren't particularly abundant in this site, but yeah, very bizarre pattern. And again, we've sampled loads and loads of sites, and you just don't see these patterns at most sites because there's always sexuals present. But it's pretty clear these things can persist without sexuals, but it just kind of basically says that, you know, whatever we're doing, we really don't have a freaking clue. So hopefully I've convinced you of, you know, the diversity in these guys and the crazy things they do is a bit mind-blowing. You know, it's a really fascinating and amazing group. And they're just, everywhere you look, you find more funky, crazy stuff going on. And the more we look, the more we're going to keep finding that kind of stuff. And really, the key next steps in moving this forward is going to be aquarium breeding trials. That's really where you got to get these things, mate them, raise them a bit, genotype them, see what's going on to really understand better what's going on. And then you can bring in the chromosome stuff and the genomic stuff in conjunction with all that sort of stuff. And always looking for people to collaborate with and you know, sucker them into carp gudgeons. We've got a really good collection, both from the genomic side and from the morphological side as well. And they're a fascinating group. And with that, I'll leave you with those guys. Thank you.
Just imagining Pete's next ARC that says, why do we need to do this research national benefit statement? Because we don't know a frequency. That's why we need to do it. All right, questions?
If you don't understand any of this stuff, that's only to be expected because it's really challenging for people to wrap their brains around this crap because it's just so unusual compared to how biology normally works. Whenever I'm out in the field and talking to people and you start saying, oh, yeah, these are hemiclonal unisexuals. And like, what? And then you try and explain it. And it's kind of like, yeah, just imagine your mum and your dad mated and made a child, but its children only had, you know, one parent's genome passed on. That's how I summarize it.
Rochelle, shoot him down. Go.
Can you repeat the
Well, the
question?
aquarium.
Of how you are investigating this question. I mean, what do you say you don't know?
Well, we need to chase some funds to get these things back into captivity and do the aquarium trials. That's the next step.
Aquarium trials.
Breeding these things and actually mating different combinations and looking at the outcomes of what goes on. Yeah, those are the fundamental next steps.
Is it possible that the hemiclone only population that was in that other person's study could be the result of a dispersal sink?
A dispersal sink, is that what you said?
Yeah, it could be a sink population.
Well, they've persisted over time though. So it could be a mating sink where basically these things just happen to mate together for no other reason than that's just who they mate with. And then both genomes get lost because maybe they can't reproduce back with anything else. We really have no idea. And Dan managed to cross all of the different forms in that population, except that one. I can't remember if he just didn't have any or if the ones he had didn't breed, but all the other ones produced offspring that he could genotype, but not that one. So he got 98% to answering the question, but then the key one didn't work for some reason.
While we're running around to Ben, I'll use this microphone to ask my question. I guess the question predictably from me is, where's the ecology? Because you've got these things that are intermediate forms and you're immediately left wondering, are they slightly diversified in diet? What's going on? Has anyone played around with using something like, you know, specific stable isotopes to do dietary ordinations? Because those ordinations you put up there, you could do exactly the same thing with compound specific stable isotopes and get some sense of whether you get that same trophic relationship.
Yeah, so that I'm waiting for the right ecologist to come along, nudge, nudge, who can who really likes doing food web stuff, because these things must be doing different things to each other. And there could be, we haven't really looked at gill morphology or mouth morphology stuff in these guys, but you know, how do you pack all these things in and have them all coexist in so many places? They must be eating different stuff and doing different things. And the stable isotopes is a really key way of kind of getting at that sort of a question. So it's really one of the next bits on the ecological side of understanding this complex. That's clearly the next step to do. The thing that's a little bit tricky with that, it's fairly cheap to keep fish in captivity and hybridize them, but it's a bit more expensive doing the food web stuff. And so that needs a little bit more serious money, unfortunately, but if we can ever sucker anybody to funding it, we'll certainly do it. Ben?
Yeah, thanks, Peter. I just wanted to ask, in terms of the environments, is there anything like the habitat or between where you get the different types of the hybrids, which sort of pops out?
So every time Ben comes into my office, he's like, we need to look at turbidity in these things. Turbidity could be really important. And my answer always is, yeah, you're absolutely right. Turbidity could be playing really interesting roles in this system. I said, the problem is, as soon as you make the fish tank turbid, you can't see anything. So it's like, I don't want to mess with that. So that's my usual ongoing dismissal of Ben when he walks in my office. Is I'm not doing turbidity stuff. It's like, yeah, the turbidity thing's a really challenging thing to work with, but no doubt, there's obviously probably some visual clues these things do. The males and females do elaborate courtshipy things that could be impacted by turbidity, and turbidity could be leading to them making mistakes and not recognizing each other properly. So it's a really interesting and fascinating area that needs a bunch more work, but it's super hard. We need little trackers on the fish so we can see them in space without actually visually observing them. Bernd?
Yeah, thanks for the entertaining talk. That was good. Just speculating, obviously, it's an interesting system, but, you can see that the hemiclones have the advantage. Probably it's a bit cheaper to reproduce, so it can be more prolific, but you have a higher parasite load, so that's bad. And, the assumption you mainly pose quite often is, someone has to die out eventually, because that's kind of how evolution works. But if you think about, you have such a great area like the NDB, and you have this flexibility in the system. So couldn't it be just a very dynamic system, and everywhere you look, you see just a certain state? Suddenly the males died out because that happened there, but in another system, it's the opposite. So maybe my question is, could it be a fairly simple system, just very dynamic, and you just have lots of different images and you can't put them together because you don't know how the dynamics are working out?
So the amazing part is these systems, just based on qualitative observation, over 30 or so years, they seem surprisingly stable. It's almost anywhere you go in the Murray-Darling Basin, which is a humongous area, you get western carp gudgeon, bullhead carp gudgeon, and you get the hemiclone, HB, HX. Almost everywhere you go, they're always there. And it's normally about 20% of the population will be the hemiclones and the rest will be the other species. So in that part, it seems like they're just ubiquitous. And so whatever happens to them, they bounce back and they seem to persist. We did start a little bit of thing where we hit a few sites where we're just like, let's just catch the first 100 carp gudgeons and try and be as least selective as possible when we're picking these things out of the net. So if we pull in the net and we had, say, 80 carp gudgeons, we just kept every carp gudgeon that wasn't too small and then genotype those to get a bit of a ballpark idea of, okay, now we've got a snapshot so we can go back later in time and see, has this ratio changed or is it stable? Certainly some of the populations have been stable, some have not. I don't know if that answers your question or not.
Yeah, I can see you have too much stable.
Yeah. So I mean, it's not like you move upstream, downstream, and things change. They're all the way through these systems, and there's thousands of them. They're a super abundant species, and so it's not like you wipe them out from an area and then you've got to start over again. Unless even in big droughts, you've got to knock absolutely every piece of water out to knock these things off, and then see what recolonizes. And that would be an interesting thing to look at, but. Yeah, they're just so ubiquitous and so abundant that they just don't seem to be too perturbed by. I mean, you don't go anywhere and find bullhead carp gudgeon without the hemiclone there as well. And that's a pretty amazing thing that the two of them are nearly always found together.
Well, we should just about stop it there.
You're going to cut off the one fish guy?
One fish guy, right, Mark?
Let Mark ask his question. Mark's been fighting with this, trying to write his book thing. He says we just named all these things once he finished his book. So I've been enjoying torturing Mark.
I'm not going to try and understand half of this shit. Simple question then, why have we only got western carp gudgeon in the ACT, do you reckon? Are they something that lives better just in upland systems or what?
So that's a totally bizarre scenario as well. So yeah, so basically once you get above Burrinjuck, you only get western carp gudgeons. There's nothing else here. And that you don't find that in any other system. So you go to the headwaters of any other system, the Murray-Darling Basin, and if anything, Clunzingeri tends to drop out a little bit, at least in Victoria, less so in New South Wales. I wouldn't be slightly surprised if they're fish from Narrandera that have come in with all the hatchery stocking and stuff that's gone on. They're Murrumbidgee mitochondrial DNA type. They could well be fully native as well, and just for whatever reason, the other ones didn't get up here. But it's an interesting question as to, is it native or not native? But it quite likely probably is. There's no reason they wouldn't be here, but it's bizarre that the others are not here.
We really should stop it there. So if you can join with me again in thanking Pete for an amazing meeting.
Guess we never checked if